I just want to thank you all for taking some time out today to listen into my webinar. My name is Dr. David Kadashko. I'm a chiropractor and acupuncturist. I've been practicing for over 30 years and have been involved with low-level laser therapy or photobiomodulation for well over a decade. My background is in molecular biology, molecular genetics, and I have a strong interest in how this therapy works in the cell, as well as obviously clinically. And my goal for today's webinar is to have a little discussion around the wavelengths or the different types of colors that are used for this type of therapy. There's a lot of interesting products out there that make lots of different claims. And it gets a little confusing, I think, to the end user, whether or not you're a healthcare professional or you're not, and you're just interested in light therapy, and you start reading, and then next thing you know, you don't know what's true or what's accurate or what's fact or fiction, right? So I'm really trying to base today's discussion around, these wavelengths. I will reference research, okay? So even though, of course, I do work for Bioflex, the wavelengths that we use and others are pretty similar, but a lot of other manufacturers, a lot of other devices that you might see out there use other wavelengths or make different claims about wavelengths or what have you. And unfortunately, it just messes everything up, and we're not able to really oftentimes know what's going on. So that's my goal today. The webinars are always recorded, and as long as you register for the webinar, then you should receive an email that has a link to watch it again. That's usually sent out within a few days as well. It's also eventually posted on our website. All the webinars are posted there. They're under customer support, just in case you're looking for them. They've been moved around a little bit. So if you go to our website, bioflexlaser.com, then you'll see customer support. I think it's under BioCare, and under that you'll see an icon for webinars, and then you'll see the different webinars there as well. I did have a question before I start, and normally I answer questions at the very end, but I'll just mention that Ron had a question about some very specific parameters as to the Personal System 180. The wavelength 660, 840 nanometers, it's accurate, and the power densities he posted are also accurate as well too. So Ron, that answers your question. Okay, so let's get started. So what I'm going to talk about is initially a bit of physics, not too in depth, but just so we understand the electromagnetic spectrum and what it is we're using when we're doing laser therapy or light therapy, or really the term is photobiomodulation therapy. And then we'll really get into more about these wavelengths and photobiomodulation, what's going on, to have a better understanding of why certain wavelengths are preferentially used and why they're more clinically effective than other wavelengths. And that's my goal, is so that we're all on the same page, and we understand that, hey, if you're interested in this type of therapy and you're interested in using it on yourself, or in a clinical setting, that you make the correct choices as far as at least wavelengths go. There's lots of other choices as well. I'll reference some research, a paper in Nature specifically, which is one of the most how should I say it, most respected medical science journals out there. And then I'll just review about what is fact, what is fiction, and I'll have time for Q&A. So if you have any questions, please type them in. I'll get to those at the very end. And I do encourage questions, okay? So let's get started. So when we talk about the wavelengths used for photobiomodulation, we need to understand the properties of light, and specifically the electromagnetic radiation spectrum, which is what you see in front of you here. This is, of course, goes back to when we studied this in probably high school and maybe university, depending what you took. And if you look at it, you can see here that at the very top, it references in nanometers the wavelength. And of course, wavelength is the distance from peak to peak or trough to trough of a wave. So nanometers being into the to the negative 9, right? And so, sorry, -6. So, all right, do I have that right? -6, I think. I should know that. So if you look at these wavelengths, you can see that there is a very small portion of the electromagnetic spectrum that is visible. And if you look at the visible spectrum, you can see it starts around 400, maybe 380 or thereabouts, and that's violet. And just shorter than that is ultraviolet. And then as you get shorter and shorter, you get into X-rays and gamma rays and cosmic rays, which sounds pretty sci-fi. And those waves are very, very powerful. They pack so much energy, they can travel through, your body like X-rays, right? They can travel through lots of different things, and they have so much energy that they're considered to be ionizing, which means that they can, in the humans anyhow, and in other animals, they can break the bonds in DNA and maybe cause mutations that can accelerate or lead to cancer, for example. So we of course know X-rays and ultraviolet exposure can lead to that as well. But as you get to the longer wavelengths in the visible band, and then even longer wavelengths like infrared, microwaves, radar, radio waves, and so forth, those waves don't have as much energy. So they're considered non-ionizing forms of light, which basically means that they're not going to break covalent bonds and hence are not considered to be dangerous in that respect. So when we talk about laser therapy or photobiomodulation, the wavelengths used are in the visible spectrum and the near infrared spectrum, and specifically red and near infrared. So if we look at the red spectrum, you can see it starts around 620 plus or minus a few nanometers. And as it gets a little bit longer, it pushes up to about 700 nanometers. And sometimes that's broken down into near red and far red. So near red would be 620, 630, and far red would be pushing into 660, going up to 700 nanometers. And what's interesting here is that when you get into that far red sort of area, the 660, that has actually been established as a sweet spot for photobiomodulation. And I'll talk about a bit about that why very shortly. So that's red, okay? So there's a bit of a spectrum. Now, what I want everyone to understand is that when we're talking about light therapy and photobiomodulation or laser therapy, there's not a specific nanometer that does the trick, okay? So I'm never going to say, oh, it has to be this exact wavelength, but it's going to be a range. So, if you're in that range, you're going to be effective. As you get out of that range, you lose effectiveness until you get to the point where there's not going to be any effect. Now, infrared is a little bit different than red. It's much longer, and it also has a much wider spectrum. So infrared, near infrared is, is going to start at maybe around 7, just where the red finishes off, obviously, right? So near infrared means close to red. And so that's starting off around, I'm going to say around 700 nanometers or plus or minus 510 nanometers. Then you can see it actually starts to get longer and longer and longer. So, you're at around 750, then you're into 1,000, 2,500 nanometers. Remember, these are nanometers, and then longer and longer. So they've broken it down into near infrared, short, medium, and long infrared bandwidth, you might say, or spectral width. Now, it's interesting to note that many of you might be familiar with things like infrared saunas or infrared heating lamps. And this infrared is true, but the wavelengths there tend to be in that short wave infrared, a little bit longer. The idea there is that the energy causes heat in the body. And they've been doing some really fascinating research to show that it increases something called shock proteins, which are actually really good for your body. They're released when you're under, when you have a fever, say, because of an infection, or an infrared sauna. Now, this is not photobiomodulation, by the way. This is a different mechanism, but they're finding that intense heat for short periods of time can release these proteins that have an actual beneficial effect on the body. Now, I won't be talking about that today, but that's as an aside. So infrared saunas, they do have a therapeutic effect. It's just not photobiomodulation. So we need to be clear about that as well, because I always get asked that question. Okay, so near infrared and the wavelengths that we're going to probably be talking about, or we will be talking about, or is going to be in that spectral width of 700-ish to 1,000, but generally speaking, in around the 800 nanometers where we're most interested in. Now, what we should understand about light is that when we're trying to get light into the body to cause this reaction, it has to go through the skin. There's no ifs and ors about it. It has to penetrate through the skin. First of all, never wear clothing or bandages or anything that will block the light from getting into your skin. When you see pictures of people and devices being sold, and they have either wearing clothes or they're doing it through different things, that's complete garbage, because that is going to get in the way. It's going to reflect, absorb light. It just makes no sense. So first of all, we got to get through skin. And also remember, if you're doing photobiomodulation or laser therapy on a patient or yourself, make sure the skin is clean with respect to oils, creams, that kind of lotions, that kind of thing, because they can absorb, reflect, and reduce the effectiveness of what you're trying to do. They actually also may heat up as well, depending on wavelengths and what's in your lotion as well. So that, as an aside, just make sure that's happening as well. So when light hits the skin, you can see that a big part of that portion of the light gets reflected off. So that's a part that you could see. So if I were to get a red laser pointer and point it at my skin, I'd see bright, intense light because most of it's reflecting back into my eye, and I can see that. And that's not what we're interested in. We're interested in light that goes through the epidermis, the top surface of the skin, and then eventually making its way into the dermis and the fatty tissue, and then deeper layers, muscle and joints and so forth. But along the way, you're going to have scattered light. It's going to be transmitted right through, and a very small portion of the light will actually be absorbed by something called chromophores. Now, the general consensus, according to the research, is that up to 85% of light will be reflected off your skin, especially if you're not touching the skin. So another little piece of advice is that if you're doing laser therapy, is that you touch the diode to the skin, ensuring better contact, better penetration. If you have a device where you're moving it around or scanning it around, then you're losing a lot more light. And most research, by most I mean 95% plus of research, references contacting the skin. So again, if you're going to follow the research and the results of the research, then you're going to do what they do, which is touching the skin. So when lasers hit the skin and make their way into the skin, they lose their coherency. We're probably familiar with lasers have a coherent beam, but when you hit skin or another, object or whatever, it's going to break apart and scatter everywhere. So what happens is it loses its energy density as it penetrates into the skin and goes deeper and deeper. So essentially, you may start with 100 milliwatts at the surface of the skin, but as you go deeper and deeper, you're of course losing a significant amount of energy. Okay, so that's also important to understand. And there's limitations how much light you should be using at your skin. If you're using a laser diode, once you push 400 to 500 milliwatts, that's the thermal threshold, and you're going to start to cause heat, which is not really what you want to do. So in general, if you're using lasers, they're going to be under 500 milliwatts, and oftentimes they're 200 to 300 milliwatts. If you're using LEDs, then typically LEDs are much less powerful, which is why you would have a lot more of them over a larger surface area. The two methods using lasers or LEDs have their advantages and disadvantages, which I won't get into a lot into today's session. So let's talk about chromophores. A chromophore essentially, has to do with a molecule that absorbs light. So, we're probably familiar with plants and chlorophyll. Now, chlorophyll absorbs most wavelengths other than green, which is why leaves appear to be green. Okay, so that's what an example of a chromophore in a plant would be. If we look at the spectral absorption of chlorophyll, you can see at the very bottom here, it's barely absorbed by green, and then it's starting to get more and more absorbed by yellow, orange, and red, and then also blue, violet, as you push the opposite direction, okay, which is why it's going to appear green. Another chromophore in plants, carotenoids, we're familiar with those in the fall time when leaves turn colors. They absorb lots of blue and violet and greenish wavelengths, but then they reflect red and orange, yellow wavelengths, which is why they appear those colors in the fall time. Now, why is this important? Well, it's important with respect to chromophores in the skin, of which there are quite a few, but the main ones really we need to be concerned about for photobiomodulation are melanin produced by melanocytes in the skin and hemoglobin. Okay, so melanin is a barrier, a pigment obviously produced by melanocytes to protect our skin from damaging ultraviolet radiation. The darker our skin, the more melanin, the more protection from UV, which is obviously why you'd have darker skin if you're from an area where there's high UV around the equator. As the farther north you go or south you go, then there's less UV. Then, of course, you have less pigment because it wasn't as required as an adaptation to where you're living. So we'd look at melanin. Melanin, by the way, what you're looking at here is a graph that shows absorption of melanin, and the bottom is the wavelength. And so I stuck in a sort of a visual there so you can see, is that melanin essentially absorbs a lot more in shorter wavelengths, especially around ultraviolet, obviously, and violet and blue. And then you see it absorbs less and less as you get towards red, although it still absorbs a little bit in the red zone. Why is that important? Well, that would mean that if we shone blue light on the skin, then very little of that blue light's going to go deep to the skin because it's being absorbed almost exclusively by melanin in the surface of the skin. Okay, so right away you're thinking, okay, well, blue light is not going to be penetrate very deeply, and that in fact is the case. Now we talk about hemoglobin. So hemoglobin dominates the absorption properties a little bit deeper to the epidermis, where there's blood, and there's different layers of blood deep to the epidermis, and also into the fatty area as well too. So hemoglobin, it tends to absorb light a lot around the 410 and 540 range, so in the blue-violet zone as well, and a little bit into the red. Again, why is this important? It's important because if you're trying to get, say, blue light, and there's many advocates for using apparently blue light for photobiomodulation, you can see here that it's not going to go very deep and perhaps therefore not be very effective. So let's look at all these different factors together. If you look at hemoglobin, melanin, and even water absorption, which I haven't even got into, you can see that they're absorbing different wavelengths along that visible and infrared spectrum. Okay, so this is really important because the chromophore that instigates photobiomodulation and the healing effect of photobiomodulation lies in between these absorption zones of melanin and oxyhemoglobin and water. So first, we need to understand what's going on with photobiomodulation. I've done this quite a few times, so I'm not going to, I know probably you're all familiar with it. So I'll just kind of skip through it fairly quickly, but we just need to understand what's going on. Of course, the effect of photobiomodulation, they have determined over 30, 40 years of research, probably 50 years now, actually, is through ATP generation or energy production in mitochondria. And of course, this has to do with aerobic respiration along the inner membrane of mitochondria. In normal aerobic respiration, there's a proton gradient pump that passes protons along, and there's energy that's being transferred using oxygen, of course, and glucose from the Krebs cycle, and it culminates in the production of ATP, adenosine triphosphate from ADP, by the terminal enzyme ATP synthase. The enzyme just before that is called cytochrome C oxidase. That's one of the terminal enzymes of the electron transport chain. And it's critical in producing ATP as well. But what happens during inflammation, so you sprained your ankle. I did that last night playing squash, and it was a little sore or whatever. So there's inflammation going on. And so during this phase, the injury will end up causing increased intracellular nitric oxide, which slows down the rate of ATP production. And by doing this, the cell starts to have a difficult time undergoing metabolism. And if it's prolonged, then these cells will undergo cellular apoptosis or cellular death. And so the goal of any type of therapy or recovery is to reduce this inflammation as quickly as possible to allow the cells and tissues to recover quickly. And that's our goal, right? And so what laser therapy or photobiomodulation does is that this cytochrome C oxidase, which I will talk about very shortly, absorbs lots of light, but especially it absorbs light in the spectral width of far red and near infrared. And as it's absorbing this light, it can actually break a hydrogen bond between nitric oxide, where it's binding on little copper and iron subunits. By doing that, it actually allows that proton gradient pump to be reestablished to some degree. The more you treat, the more this reaction, positive reaction occurs. That's the photochemical reaction of photobiomodulation, and thereby helping to produce or start to increase ATP levels. And this has a whole trickle-down effect on function of the cell, which is critical in healing. So all of these mechanisms are at play. Increase ATP, indirectly increasing sodium potassium, ATPase pump, a release of nitric oxide that causes temporary vasodilation in and around the area that you're treating. It releases free radicals in the form of reactive oxygen species, and that triggers cytoprotective signaling, as well as some wavelengths can interact with calcium ion channels and increase in calcium ion channel flow. So these are all the things that are going on that leads to a reduction of inflammation, increase in mitosis, increase in immunomodulation, reduction of edema. It can even help with producing analgesic effects and helping with collagen secretion and scar tissue formation. So these are all the positive effects of laser therapy or photobiomodulation. What's really also important to understand is that what they've determined is there's this biphasic dose response that's been well established. It's a law of hormesis, which is essentially related in part, if you can think about taking a medication where low levels of medication don't do a lot when very diluted, but as that medication is increased in strength or dosaging, you get a reaction. But that can also go to a negative reaction if the dosage is too much. Similarly, with photobiomodulation, when there's no light, there's nothing going on. But as you increase the intensity of the light or the dosaging of the light, which is typically by the power, then you'll start to get this reaction, this positive photobiomodulatory photobiomodulation reaction, but it peaks out. And it peaks out because when you start to have too much energy, you get a negative feedback loop in the cell, which again, I'm not going to get into right now, but that starts to reduce the overall photobiomodulation effect. And if you have way too much energy, then you actually get inhibition, which in itself, it can block pain, which is wonderful, but it's not very good for photobiomodulation or healing. So that's, in a sense, the whole adage of, is more better. And in the case of photobiomodulation, a lot of excessive light is actually not going to do you the benefit that you think it's going to do. So you do need to have a proper dosage. And the biggest piece of advice I give both clinicians and the general public is that if you want to do this, you need to know what kind of energy is being emitted from your device. And if it's not a medical device, meaning it's not ISO certified, you have no idea if it's accurate or not, number one. And number two, that's what I mean. If you buy something off Amazon or wherever off the internet for really cheap, you have no idea what's going on, and there's no way to verify anything. And the second thing is that you want to make sure that you're getting the correct dosage to wherever you want it to go. And that you need to understand the research, you need to understand the recommendations, you need to understand what you're trying to do with it. And to be honest, that's not something you just sort of guess at. You could, but the guesses may not be very accurate, and you may not get better. So you kind of need to know what you're doing. So this chromophore cytochrome C oxidase is the target for photobiomodulation. A lot of the pioneering work done to understand what wavelengths seem to excite this enzyme was done by Tina Carew back in the 70s out of Moscow. And she determined there are very specific peaks that energize the enzyme, and they're absorbed by copper. They can also be absorbed by iron subunits within this enzyme. And if you look carefully, you'll see there's peaks in the far red and mid-red range, and peaks in the near infrared, ranges as well. So that's why when we're trying to stimulate this cytochrome C oxidase and, take down the nitric oxide and disassociate it so that the cells can become healthier, we want to choose wavelengths that's being absorbed by this enzyme. And the work has been shown, the research has been shown that these wavelengths are the most effective. Are other wavelengths effective? Well, to a lesser degree. But once you get really out of these wavelengths, then unfortunately, you're not going to really be absorbed by this enzyme. So the point to all this is that, remember earlier I said blood and hemoglobin and water, they absorb, they're chromophores, and they absorb different wavelengths. And so even though cytochrome C oxidase might absorb other wavelengths even better, the fact remains is that they're not going to be collected by cytochrome C oxidase because they're being absorbed by blood or by melanin or by water. And so if they're being absorbed by those chromophores, very little will get to cytochrome C oxidase. Okay, and that's why you want to avoid absorption by those other chromophores and maximize absorption by the target chromophore that you're interested in stimulating. So if we look at this little chart here, at the very top is penetration in millimeters. And at the bottom, you can see is wavelength and absorption spectrum of hemoglobin and melanin and water. And because of all this, you can see that as you go from blue-violet, which is at the very top left, green and yellow, you see it barely penetrates the skin, like 1 millimeter, 2 millimeters. So if you're using a yellow or an orange or a green light on you and thinking it's doing anything deep to a millimeter deep to your skin, it's not. So they don't make good wavelengths for photobiomodulation deep to the skin. But as you get into red, it starts to penetrate a little bit deeper. So it can get into the blood and deep to the epidermis and hypodermis, and even into some soft tissues like muscle, depending on where you're treating. So it can be an effective wavelength. And the most transparent wavelength you can see is just over 800. 830, 840, 810. There's arguments around those numbers, but it's in that range of probably 780 to 840, 850. In that range is what seems to allow the light to be more transparent. In other words, it can go deeper into the skin and still be absorbed by cytochrome C oxidase. So this is another little graphic showing you how, as the wavelengths are shorter, there's less penetration. As they get longer, there's greater penetration. And remember that most light entering the tissue is reflecting and scattering and doing all sorts of stuff other than what you want it to do, which is absorption by the chromophore that you're interested in. Okay, so what's the real depth of penetration? There is no specific answer here that I could say. I use a rule of thumb to say in millimeters for red, maybe up to 10 millimeters, thereabouts. And then as you get into that near infrared, then it can go a little bit deeper. Again, depending on what's in the way and other factors as well. So there is no specific way to say exactly. Now, remember, if you have a device that you can push into the skin and depress the skin inward, then you can go much deeper. So any laser device that has more of a point to it, not point like sharp point, but sort of a pointier edge, you can actually push into the tissue and you actually can drive the light even deeper. So those tend to be even more effective for the depth. Okay, so let's look at some of the research. We're running out of time. I thought I had not enough material today. Okay, so this paper is from Nature. Hey, that's a pretty respected journal, right? Now, forget about the title because it talks about retinitis pigmentosa, which, by the way, photobiomodulation is being used on an experimental basis to treat. So it's very interesting. But what I want you to understand about all this is that the referencing for this paper published in Nature, that means, folks, it is accurate. It is fact based on science, okay? That's why I drew this paper up and showed you, is because their references, their fact checks, their peer review is going to be the best out of any kind of research journal that's out there. And in it, they talk about using far red, FR, near infrared, the wavelengths I've just mentioned. These are the ones that are going to improve the bioenergetics of the cell, restoration of cellular homeostasis, and enhanced cell survival. This is all quoting from the paper, okay? So this is a little technical. I'm not going to spend a lot of time on it. But again, from the paper, it talks about how this photoacceptor molecule, cytochrome C oxidase, which is what I've been talking about, is the target. So it undergoes a conformational change, as I mentioned, when light hits it. That conformational change is when nitric oxide releases from the binding site, as I mentioned earlier. And one of the byproducts is the release of ROS, or reactive oxygen species, as well as the release of nitric oxide. And a lot of these things will lead to positive effects, including cytoprotective genes being upregulated. That just means there's proteins called stress proteins that are increased that help the cell survive against things like inflammation. Okay, so the overall effect is improvement in cell survival or healing, right? This is what we're talking about. So the reason, again, why I mentioned the paper is this. CCO, cytochrome C oxidase, has two absorption bands that I've just talked about, one in the far red and one in the near infrared spectrum. Okay, that is why you should be using these wavelengths in order to have the most profound effect when using photobiomodulation. So fact or fiction? Fact. Photobiomodulation is a photochemical reaction. It improves cellular bioenergetics that involves absorption of light by a specific chromophore, as I've mentioned, cytochrome C oxidase. Fact, in order for light to be absorbed by the target cytochrome C oxidase, it must be in its spectral absorption bandwidth and not be absorbed by other chromophores like hemoglobin, melanin, water, and so forth. Wavelengths shorter than red and longer than near infrared are absorbed by other chromophores, as we just talked about, making them ineffective for photobiomodulation. Fiction, applying light a distance from the skin is known to induce positive photobiomodulation effects. Okay, if you're interested in this at all, you've been inundated by equipment like Juve, by these LED panels that you switch on and supposedly do everything and anything that photobiomodulation claims to do. Okay, so they're using the right wavelength, congratulations. What they're not doing is touching the skin. They're not applying specific dosages to the skin, deep to the skin. Most of the light, based on physics, is reflecting off. Do they have some type of positive effect? Probably. But what they're not doing is a specific dosaging to a certain area of the body that all photobiomodulation clinical research talks about and references. So these devices are something different. They're not devices meant to treat therapeutic conditions or what have you. A red light is definitely a very, how should I say this, effective well-being light for sure. When we see red light through our eyes, it has different effects on the retina and even in our brain. But when you're using it this way, folks, it's not going to create the same effect that I've been talking about in the body. Inside the body is what we're talking about. Fiction, blue and violet are effective wavelengths for photobiomodulation. I just explained that blue and violet barely penetrate the body. And then if you're holding it away from the skin, dollars to donuts, you're getting nothing deep to the epidermis. So while it might be effective for its bactericidal effect on acne, that's not what they're oftentimes being claimed to do. They're claiming the same effects as photobiomodulation, like red or near infrared. And hey, hate to break the news to you, but the science and research does not agree with your marketing. Fact, wavelengths used in nearly all photobiomodulation research, we're talking, I don't know, 7,000 to 8,000 research papers, use 660 plus or minus, 5, 10, 15 nanometers, and 810 to 840 nanometers. By far, the most common wavelengths used. So why would you use any other wavelengths? I don't know. And the references are there as well. Fact, light used in photobiomodulation can be emitted by many different types of diodes, not just lasers, but LEDs, superluminous diode, which is a hybrid laser LED, and other sources as well. As long as the wavelength is correct and you have enough dosage and use it correctly, then in fact, you can achieve a similar effect. Fiction, a lot of light is more effective than lower dosages. And I mentioned that earlier about the biphasic dose response known to be associated with photobiomodulation. Thanks very much for tuning in, everyone. I do have a few events coming up. I do want to mention that we have some in-person events going on, and we've partnered with a company called NeuroCatch. NeuroCatch is the first cognitive brain devices being marketed now for assessing cognitive function. It's kind of like an EEG, and it's done for different reasons, for recovery and determine what's going on for post-concussion syndrome, post-stroke recovery, for sports performance, for in general brain health and neuroplasticity. We're teaming up with them to do research on post-concussion syndrome. At the same time, we'll also be doing some events with them as well. The first one is on April the 23rd. If you're in the Vancouver area or you happen to be vacationing there and you want to drop by, it's just in the morning, 9 till I think 12 or 1 o'clock in Vancouver. And we'll be demoing our equipment and NeuroCatch as well. So if you're interested, I invite you to come by and see that. We'll also be doing events in Toronto as well as Orlando with NeuroCatch, and we'll be posting those in the next day or two. I have some other webinars coming up on post-workout relief, and we have an advanced training on nerve healing, which has an early bird for the next couple of days. So if you're interested in those, you can just go to our website and sign up for them.